பயாலஜி இன் தமிழ் கோர்வை சேனல் சார்பாக அனைவருக்கும் வணக்கம். என் பெயர் கண்ணதாசன். In this video I am going to present the lesson Classical Genetics. This is chapter 2 in 12th standard botany state word book, chapter 5 12th standard CBSE book, and this is the second video. The link for the first video given in the description. You can make utilize of it. In this video we are going to learn 2.3 monohybrid cross, 2.3.1 Mendel's analytical and empirical approach, 2.3.2 test cross and 2.3.3 back cross. Along with this, we are going to learn one box information also. It is about the molecular explanation for tall and dwarf nature of Mendel's pea plants. This video also, as usual, going to be accompanied by one Google Doc form question paper, and the link for the question paper will be there in the description, and you, by clicking it, you can open the question paper and you can start answering it. It carries, as usual, 20 MCQ for both NEET and board student, and also descriptive questions. The board student can write the answer in a separate notebook and do self-correction. I hope this video will be very useful for all the students. If you like this video, please press the like button, share this video to your friends, comment on it, subscribe our Biology in Tamil Korvai channel, press the bell icon to receive the notification of our videos. Now let us enter into the video. The first concept is about the monohybrid cross. What is monohybrid cross? Monohybrid inheritance is the inheritance of a single character. It means the experiment done based on the single character, example plant height. Here I want the students to be very clear between the character, single character studied and the two traits of the single character. The two traits, by seeing the two traits, the student may be confused. Here we are studying two characters, one is tall and another one is dwarf. So why should I say it as a monohybrid cross? The tall and dwarf are the two variants of a single character. It is the stem length. So the stem length is the character that we are going to study here. So it is called as monohybrid cross. It involves the inheritance of two alleles. For each variation, there will be one allele. Capital T, the allele for the gene responsible for the plant height, and another allele, small t, it is responsible for dwarf nature of the stem length. So you please keep it in your mind. In video 1 itself, I told you allele is the alternative form of the same gene. Here, what is the gene? The gene responsible to produce stem length. It present in the two alternative forms. One is capital T and another one is small t. So these are the two alleles of the stem length gene. Understood? When the F1 generation was selfed, Mendel noticed that he counted the number of plants produced by the F1 self-pollination. How many tall plants he counted? 787 out of 1064 F2 plants were tall. So this is one of the success for Mendel. 277 of 1064 were dwarf. The dwarf trait disappeared in the F1 generation. The F1 generation only disappeared, students, and it reappeared in the F2 generation. So 787 out of 1064 tall and 277 out of 1064 dwarf. And here only we are going to learn about the genotype and phenotype. This is the place where it is given in our book. What is genotype? The genetic constitution of an individual with respect to one character is called as genotype. Say example, capital T, always an individual carries a pair of allele with respect to a character. So if the pair of allele or capital T or capital T small or small t, so we are expressing the genetic nature of the particular character or particular variation in terms of symbols. That is called as genotype. So genotype can be expressed in the form of symbols. The term phenotype refers to the observable characteristic of an organism. It can be expressed by words like tall and dwarf. So this is called as phenotype and genotype. This can be asked in 3-mark question. Please take note of it in your notebook. Punnett square. In a genetic cross, the genotype and phenotype of offspring resulting from combining gametes. These are the gametes. So these are the gametes of the male side, these are the gametes of the female side. So during fertilization, they join together. And the Punnett square, named after a British geneticist Reginald C. Punnett. This is used to express all the possible combinations of these gametes through fertilization. So all the possible genotype which could be present in the next generation or in the progenies. That is the use of the Punnett square. It is a graphical representation to calculate the probability of all possible genotype, which I already told you, of offsprings in a genetic cross. So this can be asked in one 3-mark question. Write note on Punnett square. So again I am telling you, it is the graphical representation. That is the key word. Second key word, to calculate the probability of all possible genotypes of offsprings in a genetic cross. The law of dominance and the law of segregation give suitable explanation for monohybrid cross through Punnett square. Even the dihybrid cross, trihybrid cross, nowadays we are learning and understanding by expressing it in the form of Punnett square. Reciprocal cross. What is reciprocal cross? The core concept, I will tell you students. This concept, please don't forget. If you write this concept alone, you will be getting 1 mark in the board examination. The reversal of parental type during the experiment, that is the sex of the parents. Now coming to the concept, here we are taking the pollen from the dwarf male plant and putting it on the stigma of the tall female plant. So this is the first cross. So pollen from the male to female. In the next cross, the pollen from tall plant crossed with the stigma, that is the female gamete of the dwarf plant. Then it is called as reversal of the parental type. What for it is used? You may ask the question. Now we are crossing the male dwarf with female tall, and we are getting in the F1 generation all tall plants. Now the question arises, because we took the pollen grain from dwarf plant and female gamete from tall plant, all the plants are tall. So what we are doing is, in the second experiment, we are taking the pollen grain from the tall plant and putting it on the stigma of the dwarf plant. Now we are just reversing it. Now all the plants, once again, tall plants. Now what we could understand from this is, the sex is not having any influence in the expression of the character, the tall and dwarf. In both the ways, the progenies are tall. So we could say the reciprocal cross, by the reciprocal cross, we are able to say the sex is not having any influence over the expression of the tall and dwarf. In one experiment, the tall pea plants were pollinated with the pollens from the true breeding dwarf plants. True breeding dwarf plants. What is true breeding dwarf plant? True breeding. In the video 1, I told you, for many generations, if self-pollination done with respect to one character, so we will be getting only that particular trait for n number of generation. It is pure for the particular trait or particular variation. That is called as true breeding varieties. So these two breeding varieties are crossed, then we are getting the tall plant. In this, the reversal take place. The pollen from tall plant was used to pollinate the dwarf pea plant, which gave only tall plant for Mendel. The result was the same. All the plants of the progenies were tall. What is reciprocal cross? What is its inference? This type of question, what is its inference? This type of question can be asked in 3 marks. So the tall female crossed with dwarf male. In the next cross, it is the reversal, tall male crossed with dwarf female. Matings are done in both ways, which are called reciprocal crosses. The result of the reciprocal crosses are the same. So, what is the inference? It was concluded that the trait is not sex dependent, not influenced by the sex. The results of Mendel's monohybrid crosses were not sex dependent. So, this could be the inference. So, this may be asked in the NEET also. What is the correct inference for the Mendel's monohybrid reciprocal cross or reciprocal cross in general? Monohybrid cross. Here, this is the parental plant. The parental plant or the true breeding tall plant, capital T, true breeding dwarf plant, small t, and this is the dwarf. Once they are crossed, you just see these are the gametes expressed through Punnett square, capital T, same type of gamete from tall plant, and small t, same type of gamete from dwarf plant. And in the F1 generation, all plants are selfed. The F1 generation, all plants are selfed. Now here, all the plants are in the F1 generation are tall plants, right? All of them, you see, in the heterozygous condition, they are expressing tall, so it is called as heterozygous dominant trait, dominant variation. Are you clear? All of them are heterozygous. Okay, and here the dwarf is not expressed. And here the F2 generation. What is F2 generation? How we are able to get the F2 generation? How did Mendel get F2 generation? By self-pollinating the F1 plants. Self-pollinating. So, capital T, small t from the female side and capital T, small t from the male side. So, these are the gametes going to be fertilized. So, through Punnett square, all the possible fertilization expressed here, capital T with small t, and in the same way other side also. And here we could see one is homozygous tall plant. It means 25% homozygous tall plant. And here you see heterozygous tall plant, capital T, small t. So 50% heterozygous tall plant, and again 25% homozygous dwarf plant. Small t is genotype. So with respect to phenotype, 3 plants, okay, 3 plants out of 4 are tall plants, and 1 plant out of 4 is dwarf plant. So the ratio become 3:1. With respect to genotype, the ratio become 1:2:1, because this is the homozygous tall plant and these are the heterozygous tall plant and this is the homozygous dwarf plant. So the ratio will become 1:2:1. The gene for plant height has 2 alleles, tall capital T, dwarf small t. The phenotypic and genotypic analysis of the crosses has been shown by checkerboard method or by fork-line method. Either the checkerboard method, which we have already learnt, Punnett square, or fork-line method. And here I brought it for your understanding what is fork-line method. So this can be asked in the NEET, but not in the board examination, because board exam this type of fork-line method is not given in the state board books 12th standard. Okay, so in CBSE this may be given and you may be asked what type of method it is. This is the fork-line method. Here you see capital T, small t crossed with this is the F1 plant. You just imagine this is the female and this is the F1 male. Okay, so with respect to female, it is going to be fertilized. Capital T is going to be fertilized, capital T, small t, and male is going to be fertilized, small t going to be fertilized by capital T and small t. So we'll be getting 1/4, 1/4, 1/4. These two are the heterozygous tall plant. So it is 1/2 and small t 1/4. Right? So this is called as fork-line method. Now we are going to learn Mendel's analytical and empirical approach. So this is one of the major reasons why Mendel was very successful in giving explanation for the heredity and variation. That is the reason why is considered as the father of genetics. Now let us see what is his analytical and empirical approach. Mendel chose two contrasting traits, tall and dwarf, for each character. How many characters he studied? 7 characters he studied. So every time when in all the 7 characters he did monohybrid cross. So it seemed logical that two distinct factors exist. It means two alternative form of the gene exist. That is the way in which we have to understand two alternative form of the gene. What is it? Allele. In F1, the recessive trait and its factors do not disappear. In F1, the recessive trait and its factor do not disappear. But the phenotype is not expressed. Phenotypically, it is disappeared. But the factors, it means genes or alleles, or just stay quietly inside the cells, and they are hidden or masked only to reappear in 1/4 of the F2 generation. That is 3:1 ratio, that 1 stand for dwarf. He concluded that all dwarf alleles of F1 heterozygote segregate randomly into gametes. The F1 generation, you know very well, the genotype capital T small t, and they randomly segregate from each other. So what he tried to say here is 50% of the gamete totally produced carrying capital T and 50% of the gamete carrying small t. So totally a plant produce 100 gametes, in which that is from male side or female side. In both side it is applicable. Okay, in male side 50% of the gamete will be carrying capital T and 50% of the gamete will be carrying small t. So that is what we have to understand from segregate randomly into gametes. Based on this only the law of segregation put forward by Mendel. Mendel got 3:1 ratio in F2 between the dominant and recessive trait. Dominant 3, recessive trait 1. That is what we learnt already through checkerboard. He was the first scientist to use this type of quantitative analysis. He counted the number of seeds producing tall plant and he counted number of seeds producing dwarf plants. So quantitative analysis, it is his analytical approach. Quantitative analysis. But don't forget to remember it is the qualitative inheritance. In video 1 already we learnt discontinuous variation and continuous variation. Mendel studied discontinuous variation. It is also known as qualitative inheritance. So these two words you should not be having any confusion. Analysis is quantitative analysis, but with respect to the inheritance, it is qualitative inheritance. Understood? In a biological experiment, Mendel's analytical and empirical approach, this and extra knowledge, the small extra knowledge, it will go a long way to answer critical question in NEET and AIMS. What extra knowledge? Here you see it is not perfectly 787 divided by 1064. It gives the percentage 73.966, not 75. 75 out of 100 is 3. It means 3/4. Understand? So instead of 75, Mendel got by counting the seeds only 73.966. In the same way, if you take the dwarf plant 277 out of 1064, so the percentage become 26.03, not 25%, but still the entire scientific community accepts the 3:1 ratio, right? For this, I put one video how the fraction is rounded off. So please visit our video on chi-square test. So this is through chi-square test. Those students who are interested can go through this video and understand how scientifically this can be rounded off, and scientific community is able to accept it as a truth or factor. Okay. Mendel's analytical approach is truly an outstanding scientific achievement, students. His meticulous work and precisely executed breeding experiment. See, from Mendel's experiment, we have to learn how we should learn. The technique of learning, the technique of preparing for an examination, we could learn from Mendel's experimental methodology. His meticulous work, he kept record of each and every cross he did and precisely executed experiment like emasculation, removing the anthers. Okay, why it is done? In order to show that 100% the cell pollination is not taking place. So precisely executed experiment in those days, it is really unimaginable. Propose the discrete particulate units. Before we discovered chromosome, before we discovered DNA, he told a discrete particulate unit exists for every trait, like capital T symbolized it, small t symbolized it, okay, of heredity are present and they are transmitted from one generation to another. Excellent work, excellent uh, prediction, yes, but he did not see, yes, segregation take place, this gene is separating during the gamete formation, but still he is able to understand that discrete particulate units are able to segregate during the gamete formation of the particular plant, especially in F1 generation. Now, these discrete particle units are called as genes. After we discovered the DNA and the genes, we understood that Mendel was correct in those days. Mendel's experiments were well planned. That's what I want you to understand. To determine the relationship which governed hereditary traits. This rationale is called an empirical approach. Law that were arrived from an empirical approach is known as empirical laws. Now the question arises, what is empirical, sir? It is verifiable. The result can be verifiable by experiment, not only by theory. This is called as empirical approach. Now we are going to learn about test cross. Definition. Before that, I would like to give some idea about test cross. Mendel, uh, he was confused. What happened to the dwarf trait in the F1 generation? And also, what is the nature of the tall plant in the F1 generation? Whether it is because he crossed the pure breeding variety, capital T, understood? And what is present in F1 generation? Whether it is capital T or capital T small t? The question arises. Mendel asked that question, whether it is capital T or capital T small t. So that only made him to do the test cross. Now we can enter into this actual concept. The test cross is crossing an individual of unknown genotype with a homozygous recessive. Unknown genotype, whether it is capital T or capital T small t. You want to know, then you can cross it with homozygous recessive plant. From the result, we are able to understand what is the parental plant exactly. Under what situation Mendel happened to do test cross? This is one of the questions. What he wanted to know through this cross? Why did he call it as a test cross? All the answers in forthcoming slides. In Mendel's monohybrid cross, all the plants are tall in F1 generation. In F2, tall and dwarf plants were in the ratio of 3:1. Mendel self-pollinated dwarf F2 plants and got only dwarf plants. F2 dwarf plants self-pollinated, he got only dwarf plants. The same plants appeared in F3 and F2 generations also. So he concluded that the genotype of dwarf was homozygous. It means small t. The genotype of tall plant, capital T or capital T small t from F1 and F2 cannot be predicted. So in the F2 generation, he was not able to predict its result. But how we can tell if a tall plant is homozygous or heterozygous? Now the question arises whether that plant is homozygous or heterozygous. To determine the genotype of tall plant, Mendel crossed the plants from F2 with the homozygous recessive dwarf plant. So this is the F2 tall plant. He crossed it with the homozygous recessive dwarf parental plant. So he called it as test cross because he wanted to test what is the genotypic nature of the F1 tall plant. Though it is tall plant, whether it is having the genotype capital T or capital T small t. So that is the reason why it is called as test cross. The progenies of the test cross can be easily analyzed to predict the genotype of the plant or the test organism. We are able to understand its progenies. It means the progenies of the F1 generation can be understood by this cross. Thus, in a typical test cross, an organism, pea plant, showing dominant phenotype whose genotype is to be determined. So that is the objective of the test cross, is crossed with the recessive parent instead of self-crossing it. So we are going back. One way it is also called as back cross. We are going back and crossing it with the homozygous recessive parent. Under what situation Mendel happened to do test cross, or what he wanted to know through this cross, why did he call it as test cross? All these questions are answered. For all these three questions, only one answer. Test cross is used to identify whether an individual is homozygous or heterozygous for dominant character. So this is the ultimate answer for all these three questions, because you was not clear about its nature of genotype, right? That is the situation, and you wanted to know through this cross its genotype. You wanted to know, and why he called it as test cross, because he tested the F1 plant, so it is called as test cross. So only one answer for all these questions. If heterozygous tall cross, capital T small t crossed with small t. Now heterozygous tall test cross, this one. So in this heterozygous tall test cross, what we are able to obtain is 50% dwarf plant, 50% dwarf plant and 50% tall plant. We are able to get 50% tall plant. So if the test cross ratio is 1:1, that is the F1 plant or the plant with unknown genotype is heterozygous. Make it very clear. If the ratio is 1:1, okay, phenotypic ratio is 1:1. Even the genotypic ratio is also also 1:1 only. So if the phenotypic ratio is 1:1 in the test class, the unknown genotype is heterozygous dominant. If it is that is, if all the plants are tall plants, if the all plants are tall plant, okay, and no dwarf plant appear, then the test cross, in the test cross, the unknown genotype is homozygous tall plant, right? Homozygous tall plant. Okay, so that is why the way we are able to understand its nature of genotype. Now we are going to learn about backcross. As the name implies, backcross, we are crossing the F1 plant with either with dominant parent or with recessive parent. Then it is called as backcross. There are two types of backcross. One is dominant backcross. This is the dominant backcross, and this is the recessive backcross. It involves the cross between the F1 offspring with either of the two parents. So this is exactly the backcross, student. The F1 tall plant crossed with dominant parent means dominant backcross. If it is crossed with recessive parent means recessive backcross. So the same thing. Okay, in dominant backcross, all F2 develop dominant character. So all F2 develop dominant character. Of course, genotypically 1:1 ratio, but phenotypically 1:0. Phenotypically all tall plants, but genotypically, okay, 50% of them carrying capital T, capital T, capital T, capital T. Likewise, the 50% carrying the heterozygous nature, capital T, small t. No recessive individuals obtained in the progeny. Then it is called as dominant backcross. Recessive backcross. This is what I told you already, a type of test cross. Understand? Because we are able to understand the nature of this unknown genotype. So here we are able to get 1:1 ratio. It means 50% tall plant and 50% dwarf plant. So equal proportion, tall and dwarf appear in equal proportion, and this cross specified as test cross. So both dominant backcross and recessive backcross I gave here. Molecular explanation for tall and dwarf nature of the Mendel's pea plants. Molecular explanation. Now we are able to understand, explain why in the pea plant one plant is tall and one plant is dwarf. And here molecular characterization of Mendel's gene for plant height. The reason for this difference in plant height is due to the following fact. What is the fact? It is due to the gibberellins. And you know very well about gibberellin. Gibberellin is a type of plant hormone. It is coming under the line of auxin, gibberellin, cytokinins, ethylene, etc. Understand? So the gibberellin is present means we learnt in the plant physiology itself. It elongate the stem. It elongate the stem. The stem become long. Even the cabbage become 2 meter height because of the application of gibberellins. So gibberellin is responsible to increase the stem length. So in the tall plant, gibberellin is produced in large quantity. The dwarf plant, the gibberellin is produced in a small quantity. So that is what we are able to understand here through this molecular explanation. Now let us go through the theory. The cells of the pea plants have the ability to convert a precursor molecule of gibberellins. So this is the precursor molecule of the gibberellin, you imagine, okay, into an active form. So gibberellin active is produced. So from gibberellin inactive, gibberellin active is produced with the help of an enzyme. With the help of a functional enzyme, it is called as GA1. This GA1 is produced from an allele which is symbolized as capital L small e, Lee. Lee allele, capital L small e, Lee allele code for functional enzyme GA1, right? So this GA1 enzyme converts the gibberellin precursor molecule to active gibberellin. So the plant becomes tall. Tall pea plants, sorry. Tall pea plants have one allele, capital L, small e, that code for a protein, functional enzyme. And you know very well, most of the enzymes are proteins only. Very recently, they discovered even messenger RNA also acting as an enzyme. So the old concept, all enzymes are protein, become invalid now. So most of the enzymes are proteins which function normally in the gibberellin synthesis pathway and catalyzes the formation of gibberellin GA1. So gibberellin precursor molecule is converted to GA1 active gibberellins with the help of the enzyme, functional enzyme GA1. It is arising from the allele capital L small e. The allele dominant even if it is 2 LE capital L E capital L E. So this is the dominant nature. You assume capital T, or if it is present in a single, you say this is the dominant and this is recessive, small l small e. So this is not producing that enzyme. Very clear, this only alone producing enzyme, still the plant become tall. That is what you should understand. Even in the heterozygous condition, the enzyme produced and make the plant to become tall. So in both this genotypic condition, gibberellin precursor molecule is converted into active gibberellins, so it elongates the stem. Dwarf pea plants have two recessive alleles, small l e, small l small e. So these are the two recessive alleles. Now you just compare it with small t, which code for non-functional protein. That enzyme is not functional, hence they are dwarf in nature. So the precursor molecule is there. So the question can be asked whether the gibberellin precursor will be there in dwarf plant. Precursor gibberellin is there, but it is not produced by, or it is not converted into active form of gibberellins because the enzyme is absent. You see, small e allele code for non-functional enzyme. So that is what you have to understand. So gene for plant height in peas, tall pea plant with either capital L small e, it means capital T, or capital T small t, it means capital L small e. Are you clear? So 50, that is only one gene is active in dominant condition. That is more than sufficient to produce the sufficient quantity of the enzyme to convert the gibberellin precursor into active gibberellins. But in dwarf pea plant, small l small e, comparable with small t, so no active enzyme is produced. So the gibberellin precursor is not converted into active gibberellin molecule. So the overall experiment given in the flow chart here, right? Capital L, small e, responsible to produce the enzyme GA1. So it converts the precursor gibberellin into active gibberellin, but small e, not able to produce a functional enzyme. So it is not able to convert the precursor molecule of gibberellin into active form of the gibberellins. So this is the molecular background of the tall and dwarf nature of the Mendel's plant. So this is the end of the video, student. So summary of the video content: 2.3 monohybrid cross we learned, 2.3.1 Mendel's analytical and empirical approach, 2.3.2 test cross, and 2.3.3 back cross, and also the box information, molecular explanation for tall and dwarf nature of the Mendel's pea plants. So thank you very much for watching. Thank you.